Most people think about muscle in terms of appearance, how it looks, whether there is enough of it or too much of it, whether it is the right shape in the right places. That framing misses almost everything important about what muscle actually does. Muscle is not primarily a cosmetic tissue. It is a metabolic organ, one of the most metabolically active tissues in the body, with functions that extend far beyond movement and appearance into glucose regulation, immune function, hormonal signaling, cognitive health, and longevity. And it is an organ that most people are slowly losing without knowing it, starting earlier than they expect, with consequences that accumulate quietly for decades before becoming obvious. Understanding what muscle actually does changes the motivation for maintaining it entirely. Not aesthetics, not athletic performance, biology. Skeletal muscle is the largest single site of glucose disposal in the human body. At rest, muscle accounts for approximately 20 to 30% of total glucose uptake. During physical activity, that proportion increases dramatically. Contracting muscle can absorb glucose at rates up to 50 times higher than resting muscle, primarily through a mechanism that operates independently of insulin. This is the first and most immediate metabolic function of muscle mass. It is the primary buffer against excess blood glucose. When you eat carbohydrates and blood glucose rises, muscle tissue is the destination for the majority of that glucose. The larger and more metabolically active your muscle mass, the more efficiently your body can clear post-meal glucose spikes, the lower your average insulin levels, and the more stable your blood glucose across the day. This relationship has direct implications for metabolic health. Insulin resistance, the condition in which cells stop responding normally to insulin signals, develops partly as a function of inadequate muscle mass. A person with low muscle mass has less glucose disposal capacity. Blood glucose rises higher after meals and stays elevated longer. The pancreas produces more insulin to compensate. Over time, cells become less responsive to the elevated insulin signal. Insulin resistance develops, and with it, the cascade of metabolic consequences: increased fat storage, elevated triglycerides, chronic inflammation, and eventually type 2 diabetes that follow from sustained hyperinsulinemia. This is why muscle mass is one of the strongest predictors of insulin sensitivity independent of body weight and body fat percentage. Two people at the same weight and body fat level will have meaningfully different metabolic profiles if one has significantly more muscle mass than the other. The one with more muscle has greater glucose disposal capacity, lower baseline insulin, and substantially reduced risk of metabolic disease. It is also why resistance training, the primary stimulus for muscle maintenance and growth, produces metabolic benefits that extend far beyond the calories burned during the session itself. A well-designed resistance training program increases muscle mass, which increases resting metabolic rate and glucose disposal capacity on a continuous basis, not just during exercise, but 24 hours a day, 7 days a week, for as long as that muscle mass is maintained. Sarcopenia is the medical term for the age-related loss of muscle mass and strength, and it is one of the most significant and least discussed processes in human aging. Muscle loss begins earlier than most people expect. After the age of approximately 30, skeletal muscle mass declines at a rate of roughly 3 to 5% per decade under sedentary conditions. After 50, that rate accelerates. By the time most people reach their 70s, they may have lost 30 to 40% of the muscle mass they had in their 30s, and in many cases, significantly more. The consequences of this loss are not limited to weakness and reduced physical capacity, though those are real and significant. Sarcopenia is independently associated with increased risk of type 2 diabetes, cardiovascular disease, cognitive decline, depression, and all-cause mortality. It is associated with increased susceptibility to infection. It is a major contributor to the loss of independence in older adults. The inability to rise from a chair, to climb stairs, to recover from a fall, that dramatically reduces quality of life. And it is largely preventable and to a significant degree reversible through resistance exercise. The research on resistance training in older adults is among the most consistently positive in exercise science. Studies on people in their 60s, 70s, and 80s who begin resistance training programs show significant increases in muscle mass, strength, and function within weeks to months. The muscle tissue of older adults responds to resistance training, more slowly than that of younger adults, but the response is real and the functional improvements are clinically meaningful. This does not mean the loss is effortless to reverse or that the window for action has no limits. It means that the timeline for beginning is not fixed, and that beginning later is meaningfully better than not beginning. Muscle is not metabolically silent between exercise sessions. It produces and releases signaling molecules called myokines, proteins that act as hormones, communicating between muscle tissue and other organ systems to coordinate physiological responses across the body. The discovery of myokines has significantly changed how researchers understand the relationship between physical activity and health. The benefits of exercise, reduced inflammation, improved immune function, better cognitive performance, enhanced mood, reduced cancer risk, are not fully explained by the direct mechanical effects of movement or even by changes in cardiovascular fitness. A significant portion of these benefits are mediated by the myokines that muscle produces in response to contraction. Interleukin-6, released from contracting muscle during exercise, has anti-inflammatory effects at the systemic level. Paradoxically, given that the same molecule produced by immune cells in the context of infection or injury has pro-inflammatory effects. Muscle-derived interleukin-6 suppresses tumor necrosis factor alpha and interleukin-1 beta, two of the primary drivers of chronic, low-grade inflammation that underlies cardiovascular disease, metabolic syndrome, and several cancers. Irisin, another myokine, is released during resistance and aerobic exercise and has been shown to cross the blood-brain barrier. In the brain, irisin increases the production of BDNF, the same brain-derived neurotrophic factor that supports neuronal growth, synaptic plasticity, learning, and memory. The relationship between exercise, irisin, and BDNF is one of the primary mechanisms through which physical activity produces cognitive benefits, including the well-documented associations between regular exercise and reduced risk of Alzheimer's disease and cognitive decline. A 2022 study published in Nature Metabolism found that irisin levels in cerebrospinal fluid were significantly lower in Alzheimer's patients compared to cognitively normal controls, and that restoring irisin levels in animal models of Alzheimer's disease improved memory function. The implication is direct. Muscle-derived signaling molecules are not peripheral to brain health. They are part of the biological maintenance system that keeps the brain functioning well. This myokine communication system explains why the cognitive benefits of resistance training are distinct from and additive to those of aerobic exercise. Both produce irisin and BDNF. Both reduce inflammation. Both improve insulin sensitivity in the brain. But resistance training produces a pattern of myokine release and hormonal response, including growth hormone and testosterone stimulation, that has specific effects on neural tissue that aerobic exercise does not fully replicate. The immune system connection to muscle mass is less commonly discussed, but mechanistically significant. Skeletal muscle is a major reservoir of amino acids, the building blocks of proteins, including the proteins that the immune system requires to mount an effective response to infection or injury. When the immune system needs to produce antibodies, cytokines, and immune cells rapidly in response to a pathogen, it draws on amino acid pools throughout the body. Muscle is the primary reserve. In people with low muscle mass, either from sarcopenia, chronic illness, or prolonged inactivity, this reserve is diminished. The immune system's ability to mount a rapid, robust response to infection is impaired not because of any direct defect in immune function, but because the raw materials available for producing immune proteins are insufficient. This is one of the mechanisms behind the well-documented association between low muscle mass and worse outcomes from acute illness, including infections, surgical procedures, and trauma. A person with greater muscle mass has more metabolic reserve to support the demands of an acute immune response. A person with low muscle mass is more vulnerable not just to the illness itself, but to the metabolic cost of fighting it. The COVID-19 pandemic generated a significant body of data on this relationship. Multiple studies found that patients with lower muscle mass, measured by imaging in hospitalized patients, had significantly worse outcomes, longer ICU stays, higher rates of mechanical ventilation, and higher mortality, independent of age, body weight, and other comorbidities. The muscle mass, it turned out, was not incidental to recovery. It was a resource. Here's what the research supports practically. Resistance training. Lifting weights, body weight exercises, resistance bands, or any training that places mechanical load on muscle tissue is the primary stimulus for maintaining and building muscle mass at any age. Cardiovascular exercise is valuable for cardiovascular health and produces some myokine benefits, but it does not produce the same muscle-preserving stimulus as resistance training. Both are beneficial. Neither fully substitutes for the other. The minimum effective dose for muscle maintenance in adults is lower than most people expect. 2 sessions per week of compound resistance exercises, movements that engage multiple muscle groups simultaneously, such as squats, deadlifts, rows, and presses, are sufficient to maintain muscle mass and produce meaningful improvements in strength and metabolic function. More is generally better, up to a point of recovery capacity, but 2 sessions per week represents a threshold below which significant muscle loss occurs over time and above which meaningful preservation occurs. Protein intake is the dietary variable most directly relevant to muscle maintenance. Muscle protein synthesis, the process by which muscle tissue repairs and builds in response to training, requires adequate dietary protein as a substrate. The research on optimal protein intake for muscle maintenance and growth consistently suggests that most people, particularly older adults, consume less protein than is needed to support muscle health. Current evidence supports a target of approximately 1.6 to 2.2 grams of protein per kilogram of body weight per day for people engaged in resistance training, with older adults benefiting from the higher end of this range due to reduced anabolic sensitivity to dietary protein. The distribution of protein across meals matters. Muscle protein synthesis is maximized when protein is consumed in adequate amounts at each meal, rather than concentrated in 1 or 2 meals. A target of approximately 30 to 40 grams of high-quality protein per meal, from animal sources, legumes, or high-quality plant proteins, appears to maximally stimulate muscle protein synthesis in most adults. Here's the framing that changes how most people think about this. Muscle is not optional equipment for athletes and people concerned with their appearance. It is a functional organ whose decline has measurable consequences for glucose metabolism, immune function, cognitive health, hormonal balance, and lifespan. Its maintenance requires deliberate effort because the modern environment, sedentary work, readily available processed food, disrupted sleep, does not provide the stimuli that maintain it naturally. The person who lifts weights twice a week is not primarily optimizing for how they look. They are maintaining the organ that buffers their blood glucose, supplies their immune system with metabolic reserves, sends irisin to their brain to support neuronal health, and provides the physical capacity that determines whether aging is a gradual reduction in capacity or a sudden cliff. Sarcopenia is not inevitable. It is the default outcome of inactivity. The alternative outcome is available to anyone willing to apply a mechanical load to their muscles with sufficient regularity to provide the maintenance stimulus that muscle tissue requires to remain. 2 sessions per week, compound movements, adequate protein. That is the minimum. The biology does the rest. If this changed how you think about building muscle as something other than vanity, hit the like button. Share it with someone who thinks the gym is only for people who care about how they look. And subscribe for more content like this.